Today, FDA approvals of an HPV vaccine and a next-generation sequencing assay, a priority review designation in multiple myeloma, and an ODAC recommendation for a biosimilar. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved the HPV vaccine Gardasil 9 for use in males and females aged 9 through 45 years. Gardasil 9 has been shown to prevent cancers and other diseases caused by 9 types of HPV: 6, 11, 16, 18, 31, 33, 45, 52, and 58. The FDA initially approved Gardasil 9 in 2014 for use in males and females aged 9 through 26 years. Gardasil, the predecessor to Gardasil 9, was approved by the FDA in 2006. Gardasil is no longer distributed in the United States. However, efficacy data for Gardasil can be extrapolated to Gardasil 9, since the two vaccines are similarly manufactured, and Gardasil targets 4 of the 9 HPV types covered by Gardasil 9. In a large trial of women aged 27 to 45, Gardasil demonstrated an efficacy rate of 88% in preventing persistent infection, genital warts, vulvar and vaginal precancerous lesions, cervical precancerous lesions, and cervical cancer related to HPV 6, 11, 16, and 18. The study included approximately 3,200 women with an average follow-up of 3.5 years. The FDA used these data, as well as updated long-term follow-up data from the same study, as the basis for increasing the approved age up to 45 for Gardasil 9 in women. The new approval of Gardasil 9 for men aged 27 to 45 was based on inferred efficacy from the 33,200 women Gardasil trial and efficacy results for Gardasil in men aged 16 to 26. The agency also reviewed immunogenicity data from a study in which a 3-dose regimen of Gardasil over 6 months was assessed in men aged 27 to 45 years. The FDA has approved the next-generation sequencing assay clonoSEQ as a test for minimal residual disease in patients with acute lymphoblastic leukemia or multiple myeloma. The in vitro diagnostic identifies and quantifies gene sequences in DNA extracted from the bone marrow of patients with ALL or multiple myeloma using multiplex polymerase chain reaction and NGS. The assay detects as few as 1 tumor cell within more than 1 million healthy cells. Moreover, the clinical validity of clonoSEQ was seen in a retrospective analysis of samples collected from a clinical trial involving patients with ALL and 2 trials involving those with multiple myeloma. Researchers in these trials used the assay to assess MRD at various disease burden thresholds and determine that MRD status correlated with event-free survival. EFS was longer and EFS rates were higher for patients assessed as MRD negative. Results were similar for progression-free survival and disease-free survival among patients with multiple myeloma. The agency reviewed clonoSEQ through the de novo pre-market review pathway for new types of novel low to moderate risk devices. Additionally, the FDA established special controls clarifying expectations regarding the efficacy, accuracy, and reliability of tests using MRD to assess changes in disease burden during and following therapy. In multiple myeloma, the FDA has granted a priority review to a new drug application for selinexor for the treatment of patients with penta-refractory disease. Selinexor links to and inhibits XPO1, which is a nuclear export protein. The NDA is based on data from the Phase 2b STORM trial, which evaluated the combination of selinexor and dexamethasone in patients with penta-refractory multiple myeloma. Updated findings from the study show that the doublet regimen was associated with an overall response rate of 26.2% and a median overall survival of 8.6 months in patients with penta-refractory disease. Overall, patients who had any response to treatment did significantly better, with a median OS of 15.6 months. Historically, the median OS in this patient population is an estimated 3.6 months. The most common grade 3 or 4 treatment-related hematological adverse event was thrombocytopenia, followed by anemia, neutropenia, leukopenia, and lymphopenia. There was only 1 case of grade 4 anemia and no grade 4 leukopenias recorded. Under the Prescription Drug User Fee Act, the FDA is scheduled to decide on the NDA by April 6, 2019. The FDA's Oncologic Drugs Advisory Committee voted 16-0, recommending approval of the rituximab biosimilar CT-P10 for 3 of the anti-CD20 monoclonal antibodies non-Hodgkin lymphoma indications. Should the FDA agree with the panel's recommendations and grant approval to CT-P10, the biosimilar would be approved to treat adult patients with CD20-positive follicular lymphoma plus chemotherapy in the frontline setting, followed by single-agent maintenance in responders. As a monotherapy for relapsed refractory low-grade or follicular CD20-positive B-cell non-Hodgkin lymphoma, and as a single agent in patients with low-grade CD20-positive B-cell non-Hodgkin lymphoma who do not progress following first-line chemotherapy. ODAC was tasked with determining whether the data submitted in a biologics license application by Celltrion and Teva Pharmaceutical Industries, the co-developers of CT-P10, demonstrated the biosimilarity between CT-P10 and rituximab in terms of efficacy, safety, pharmacology, analytical similarity, and immunogenicity. The panel specifically considered findings from 2 randomized, double-blinded trials submitted with the BLA, both of which showed no clinically meaningful differences between CT-P10 and standard rituximab. Beyond follicular lymphoma, rituximab has approved indications for diffuse large B-cell lymphoma and chronic lymphocytic leukemia. This week, we sat down with Dr. Tanios Bakai-Saab of Mayo Clinic to discuss later lines of therapy for metastatic colorectal cancer.
Almost 90+% of the patients who progress through first line will make it to second line. And second line options will depend on what you've done in the first line. Like I said, for most US physicians, chemotherapy plus bevacizumab is their primary option. For some, on the left side, RAS wild type, BRAF wild type, HER2 non-amplified, they choose to start with an EGFR inhibitor plus chemotherapy. So let's first start with all these patients who will receive chemotherapy plus bevacizumab in the first line, regardless of their genetic or molecular subtyping. Second line, you switch the chemo. So if you start with FOLFIRI, you go to FOLFOX. If you start with FOLFOX, you switch to FOLFIRI. And then you have the option of continuation of the VEGF inhibitor. So bevacizumab, you continue bevacizumab to the second line. I think on the right side, that makes sense. On the left side, however, that only makes sense in those patients who have RAS mutations or HER2 amplifications. For the BRAF mutations, that's a particular case. For those patients who have RAS wild type, BRAF wild type, and are HER2 non-amplified, if you started with chemotherapy plus bevacizumab, your second line has to be an EGFR inhibitor plus chemo. And the reason for that is because we know that EGFR inhibitors work pretty well in this group of patients, and I wouldn't want to wait to third line before I initiate that treatment. So for those patients, you can use EGFR in the first line, then possibly VEGF inhibitors, so bevacizumab in the second line plus chemo. If you started with bevacizumab, then an EGFR inhibitor in the second line. For the BRAF V600E, we try to have them either on a clinical trial with a RAF plus MEK inhibitor plus minus EGFR inhibitor, or now the NCCN guidelines do list this as an option, which we sometimes consider utilizing. On the right side, it's Bev, regardless of the mutational status, and then the third line would be regorafenib, or TAS-102 would be an acceptable option. On the left side, again, regorafenib would be your third line option, or TAS-102 again would be an acceptable option. So what % of patients actually make it to the third line is we think up to 50 to 60% of patients who started first line will end up in third line. When you get to fourth line, you lose even more of those patients. It'll end up being about 25 to 30% of the patients that end up in fourth line. So if you use regorafenib, which I do in my clinic, in the third line, then TAS-102 follows. If you use TAS-102 in the third line, then regorafenib would follow. For the case of the HER2 amplified patients, second line, usually we shift them to one of our clinical trials with a drug called tucatinib plus trastuzumab. That's a study that we have in our site and a number of other sites nationally. And that essentially bypasses the need for an EGFR inhibitor, which we know from other from the MD Anderson experience, at least in 2 cohorts, that those HER2 amplified patients do not seem to have benefit from EGFR inhibitors. That kind of makes sense understanding the interplay between EGFR and HER2. So it's not surprising to see that if there's a benefit, it's minimal. Now, a very important question is, as we move beyond second line to third line, is what do you do with those patients in the third, fourth, or fifth line on the right side? Do we ever expose them to EGFR inhibitors? The answer is, although the data is very weak for EGFR inhibitors, even though those that are RAS wild type, I still think there's a place for them. There's a study that looked at essentially EGFR inhibitor cetuximab versus best supportive care that suggested that in the later lines of therapy, there may still be a little bit of a survival advantage for cetuximab versus best supportive care. There was no PFS advantage, but overall survival advantage. And that could essentially tell us that there may still be a role for EGFR inhibitors in later lines of therapy in patients on the right side. I usually use it after regorafenib and possibly after TAS-102 if I run out of options. On the left side, in the HER2 amplified patients, the RAS wild type HER2 amplified patients, I get back to these EGFR inhibitors sometimes around between the third and the fifth line of therapy. There are nuances, of course, to all these. The MSI high patients go on a PD-1 inhibitor and try actually to move it to first line if the insurance will allows me, but I wouldn't wait beyond the second line. That's for 4% of the patients. So it's a landscape that keeps on shifting, but overall, parts of it remained quite stable.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.